Plant density, counting methods. Density is defined as the number of individuals per unit area. Density reflects the closeness of individuals to one another, and counting methods are among the most commonly used approaches to estimating density. Counting methods are plot-based, meaning that they occur in areas that have edges. Common types of plots are quadrats or frames. These tend to be somewhat small, at least can be carried out into the field, usually constructed of some type of a hard material such as steel, aluminum, PVC pipe, and they can be in different shapes and sizes. So the shapes that we commonly see are square, rectangular, or circular, and as I mentioned, the sizes are generally small enough that someone can easily carry them and deploy them in the field. If we need to use a plot that is larger than, say, 1 meter squared or 1.5 meters squared, then we will just use plots that we establish in the field using transect tapes, usually staking the corners of the plots, and then sometimes leaving the transect tapes in place to help us delineate the boundary of the plot. Often when we're measuring density, we want to use very long plots that have a relatively narrow width, and these are called belt transects. These are established using transect tapes, and then by walking a swath and measuring the distance of that swath or the width of that swath, then counting the plants inside the belt transect. Counting methods, in order to determine density, have to be calculated in a specific way. Remember, the units of density are the number of plants per unit area. And so in this example, we're looking at this small community of plants, and we're going to estimate the density of pink daisies to the nearest meter squared. We're going to do this using 3 quarter-meter squared quadrats, and we count the number of quadrats, the number of pink daisies inside the quadrats. From this count, we see that we've got 2 quadrats that include 1 plant and 1 quadrat that doesn't have any pink daisies in it. The calculation is to sum the total number of plants and divide that by the total number of quadrats observed. That includes the empty quadrats, and that calculates out to 0.66 plants per quadrat. But the units that we want to report are in terms of the meter squared, so we need to convert that number to number of plants per meter squared, and that equals approximately 2.7 plants per meter squared. For any type of measurement that we do, we need to clearly define ground rules. An important ground rule for counting methods is to identify the counting unit. Oftentimes we're interested in individuals, such as this individual bunch grass. In some cases, it's difficult to determine what is an individual. This is true for plants that are clonal or that reproduce vegetatively below or above ground, or for plants that are very closely spaced together. It may not be necessary, depending on what your management objectives are, to identify the individual plant, and it may be sufficient simply to count the number of plant parts, such as stems, tillers, or inflorescences. Another important ground rule that pertains to counting units is what determines whether a plant is counted as in the plot. The most common rule is that plants must be rooted inside the plot. So merely having canopy that overhangs does not qualify the plant as being counted. Sometimes, though, there are plants in the environment that we are interested in being able to quantify, yet they may, because of their size and the way that they grow, their life form, it may be unlikely that you would capture the base of the plant inside the plot. And in this case, we may consider overhanging canopy as qualifying a plant for being included in the plot. This is something that we determine for selected species, and we must very carefully record which species are included merely for having overhanging canopy. Another thing that's important is when we decide whether the base of a plant is included in or out. Quadrats and plots have edges, and so that means that at these boundaries we have to make decisions about whether the plant is included in or out. So our decisions are, do we include the plant if the stem of the plant touches the edge of the quadrat, such as here or here? Do we decide that it won't be counted if it has less than 50% in, if it must have more than 50% of the stem in? Or what do we do in the case of having exactly half and exactly half of the stem in and half of the stem out? Well, in this case, we want to establish some decision rules. And one very commonly used approach is to divide your quadrat frame into half. So you have you establish that two of the sides, say the top and the left side, if you have to make a decision about a plant, those plants are included as in if they touch or cross the top or the side, the left side of the plot, and they would be excluded if they touch or cross over the right or the bottom side. Obviously, plants that have all of their stem inside would be counted as in. Another way to do this is when you have a plant that you need to make a decision about, you simply use the every other rule. So if the first plant that you have to make a decision about gets counted in, the next time you have to make a decision, that plant would be counted out. Another approach is to closely examine each individual case and then decide whether more than half of the plant is in or out. This approach is not recommended, however, because it can be quite time-consuming, and there's always the potential for observer error or a bias to be introduced. For any sampling effort that we do, our objectives are precision, accuracy, for the most efficient sampling effort. And so how do we select the right type of sampling unit? What is the proper size or shape? Well, our desired precision is important. Also, whether the plants, what the average plant size will affect the size and the shape of the quadrat that we select, and also the dispersion of plants, whether they are evenly distributed or homogeneous in the environment, if they are heterogeneously distributed or patchy and clumped, and if there is a spatial gradient. This will affect whether we choose to use elongated, different shape quadrats, and also the size of the quadrat. Let's look at that in a little more detail. If we have relatively small quadrats, we could have a number that are empty and only a few that have any plants at all. This would not lend to having good precision in our estimate. By increasing the quadrat size, we can increase the number of quadrats that have plants in them, and there will be greater similarity in the number of plants that are in the quadrats. In general, the guidelines are that you want your quadrat size to be larger than the average plant and also larger than the average space between plants. Let's consider quadrat shape. If we go from a square quadrat to more rectangular quadrats without changing the size, we find that in patchy vegetation, we can include more plants and cut across the variability of the vegetation. The general rule is that more elongated quadrats tend to cut across clumps, and that this will increase the precision in sparsely distributed vegetation. Let's take a look at the influence of size on precision. In this case, we have a population of known density, and we're comparing the results that we get just by changing the size of the quadrats. So we're using square quadrats of different sizes. The small ones are 1 meter squared, and the large ones are slightly more than twice the size at 2.25 meters squared. What we see is because we reduce the number of empty quadrats by increasing the size, we can decrease the standard deviation in our estimate, and that will result in a more precise estimate. Let's now look at the effect of shape, changing the shape on the precision. In this case, we're keeping the size the same, 1 meter squared quadrats, but we're simply elongating them. And again, what we see is that we go from a situation where we have several more empty quadrats than those that have any plants in them, to having more that have plants included inside. And this reduces the standard deviation from 1.3 to 0.4 plants per meter squared. So we've increased the precision. Now, you may say, well, what if we have plants that we're interested in that have different sizes, different types of distribution? We're still interested in all of those plants. In that case, we would use nested quadrats, and we would select the appropriate size quadrat for different species. In this example, there's the grass plants. There are far more of them, and we only need a smaller quadrat to estimate grass. We use the slightly larger quadrat to estimate the density of the daisies, and we use the largest quadrat to estimate the density of the brown thistle. So that's one application of using nested quadrats. It's very efficient to do this. Again, with nested quadrats, we use different sizes and shapes simultaneously for different species, or sometimes for different functional groups, or for different age classes. So for example, in this bottom diagram, we could use, if we were interested in seedlings, we would count seedlings only in the smaller quadrats. We would count saplings or juveniles in the belt transects, and then adults in the larger plots. So to review the general guidelines for counting methods, you always want to conduct pilot sampling so that you can determine the appropriate size and shape of the quadrat or plot for your area of interest. We generally increase the plot size if more than 20% of the plots are empty, or if the number of empty plots is greater than the number of plots with one or more plants in them. In sparse vegetation, we tend to elongate the shape of the plots, and we use nested quadrats if the species of interest require different plot sizes. Remember the importance of clearly stating your ground rules before you initiate your sampling efforts.